Get rid of that. Okay, boys and girls, today let's finish talking about chapter 7, influences on microbial growth. We left off, I believe, talking about osmotic oxygen requirements. And the last little thing that I want to say about oxygen requirements is that bacteria that require a limited amount of oxygen, so aerotolerant anaerobes, microaerophiles, obligate anaerobes, of course, they do need special culturing conditions. So what I mean by that is in the laboratory, in order to get them to grow, we need to make the conditions so there is less oxygen to zero oxygen. And there are a few ways to do that. I'd like to just run through them real quickly. The first method is to use what's called reducing media. And I usually cover this back in that chapter 3 bit, and I think I forgot this time. So reducing media is just regular old nutrient agar or trypticase soy agar. It's usually prepared in a test tube as a deep, and it has a special additive in it, and that additive is called sodium thioglycolate. Sodium thioglycolate, and you'll find the spelling of that either in, I believe, back in chapter 3. And what sodium thioglycolate does is it absorbs or binds free oxygen in the media that's dissolved in the media and binds it up so that it's not present in its free form in the tube. This allows bacteria that are anaerobes or aerotolerant or microaerophiles to, not so much microaerophiles, but aerotolerants and obligate anaerobes to be able to grow in the media, and they don't need any special chamber or environment, okay? There is a picture in your textbook. It is figure 7.9, and it is showing this reducing media and where, actually, I believe this is actually not reducing media, it's just regular media, and it shows where the bacteria grow based on their oxygen requirements, meaning the obligate anaerobes will only grow at the bottom of the tube where there's little to no oxygen, whereas the obligate aerobes will grow at the top, and the facultative anaerobes will grow all the way through the tube. Okay, so one thing you can do is prepare special media. The second thing you can do is you can put the bacteria into a chamber that reduces or eliminates the free oxygen. And there are two main methods for this. One is to use what we call it a brewer's jar. And the second method is to actually use an anaerobic incubator. So an anaerobic incubator, a brewer's jar is small, relatively small. So it would sit about this big on the table and about this tall. I mean, so from the table, right, be about this big. It's like a giant, one of those giant pretzel containers or cheese ball containers, about that size, with glass or really thick plastic. And inside of it, it has a lid that fits super tightly. And inside of it, there's usually a rack so you can stack Petri dishes. And it has, you put a chemical reaction, you add water to a chemical reaction pouch, and you put the pouch into the chamber. You load up your Petri dishes that you've streaked your anaerobes on, and then you tighten down the chamber. And what happens is the chemicals in that pouch absorb all the free oxygen inside the chamber. So it's a completely anaerobic chamber until I open the jar. So this will allow anaerobes to grow so that we can observe them. And then you would open the jar, look at the bacteria, make your notes, subculture them if you needed to, and then they're dying as soon as they are exposed to that oxygen. Very old-fashioned, we used to, sometimes in a clinical laboratory, super low budget, we would make what we called candle jars for aerotolerance. And this is not going to be on the test, but we would use old glass pickle jars, and we would stack the Petri dishes in them and put a piece of cardboard and a small stub of a candle. And when you light the candle, close the lid, just like in chemistry, when there's no oxygen, there's no flame. The candle would go out, and then that chamber would be relatively anaerobic for a day or two. Remember, bacteria grow so quickly, all you really need is a day or two in order to be able to see their growth on a Petri dish on a plate. Now, more complicated than that, so if you're working in a clinical laboratory where you're getting specimens from patients, maybe from a wound that is suspected to be infected with an anaerobe, or maybe someone has a blood infection that might be anaerobic or aerotolerant. In that case, those clinical laboratories would have an anaerobic incubator. So usually it's connected to a hood, a laminar flow hood, that would reduce the amount of oxygen in the hood itself. And then there's also a photograph in your textbook, I believe, of a woman working in the anaerobic chamber. So you can see her there, figure 7.8. She has a big plastic shield in front of her, and her hands are inside gloves where she's working without actually touching the bacteria. And then right off to what would be her left in the picture, you can see the opening of a small tabletop incubator that she's popping her cultures into. And that incubator would be fed carbon dioxide gas in a canister, again, to limit the amount of oxygen in that container, as well as sometimes very specialized ones would also have a mechanism to vacate the oxygen. So those are some methods for growing anaerobes. And why would you need that? Again, in a clinical setting, I've already described it. In an environmental setting, if we're looking at bacteria that grow in swamps, bogs, deep in the soil, places where there's very little dissolved oxygen, deep in the ocean sediments, for example, those would all be situations where if we were trying to grow those bacteria in the laboratory, we have to recreate exactly their living conditions. So we need to make it really cold or really hot, and we need to make it anaerobic or aerobic, depending on what their nutritional requirements are. All right, enough about that. pH just gets a mention as far as an environmental condition. Clearly, most bacteria grow best, especially pathogens, ones that infect humans and animals, between pH 6 to 8, because we're about pH 7. But if I'm trying to grow a bacteria, again, that lives in acid, I would have to make an acidic environment. And to do that, then I would have to either make it, like I said, acidic or basic. I'm usually using buffers in the solution, and as well as acids and bases. Your book discusses a few. We would call those bacteria that prefer acid. We call them acidophiles, love acid loving. There's also a section there on osmotic pressure. Primarily, we're talking about salt or high concentrations of various salts, not just sodium chloride, but all kinds of chemical salts. Those are referred to as halophiles, salt-loving bacteria, and some of them grow at concentrations up to maybe 25% salt. So places like the Great Salt Lake, the Dead Sea, caves, other places where there are super salty concentrations, again, if we're trying to grow them in the laboratory or we're trying to investigate under what conditions they, or let's say what conditions they spoil food, we would want to be able to grow those bacteria. Also, there are bacteria that can only live under a certain amount of pressure, and we're talking about a higher pressure than atmospheric pressure. So these mostly bacteria that live in the ocean depths, those are barophiles. And again, you would have to adjust the conditions for them so that they could grow freely. If you take a look on page 184, there is a sort of a quick association of bacteria that need other bacteria or other organisms to grow. We refer to those as associations between bacteria, or we would even say symbiotic relationships, perhaps. And there's a list there, some words that you should be able to define: mutualism, commensalism, parasitism, and those all come under the category of symbiotic organisms, meaning they're living with another organism and are somewhat dependent on its existence. And then there are non-symbiotic relationships, synergism and antagonism. These are all important and are becoming more and more important interactions as we learn about the microbiome and how many bacteria live on us and with us, and how they use byproducts of each other in order to grow and live. So again, I'm not going to be asking you specific examples of those, but you should be able to recite the definitions of those words. Okay, so there's 5 terms there: mutualism, commensalism, parasitism, synergism, and antagonism. And like I mentioned, they're in a table, in a little graphic for you at the bottom of page 184 in your textbook. I'm not going to go through each one of those. We just don't have time. Fascinating as they are. There's also then a short discussion about biofilms, right? And again, the definition, you should know the word quorum sensing. I probably won't be including a lot about biofilms in the connect, but if I do, that's the section that you should refer to. And then finally, we're on to talking about actually growth and counting microbial growth, which is 7.3, is the last section in this chapter. So the first thing that I want to mention is, and I'm going to move over to word here. Someone has been moving my setup on me. Okay, so the first thing I want to mention is I want to remind you that bacterial reproduction is not mitosis or meiosis, but is binary fission. It's accomplished by what we call binary fission. And that's because, remember, bacteria do not have organelles that they need to divide. There's no nucleus. So it's a much simpler process in that, and there's only one chromosome, so we don't need to form mitotic spindles. We don't need to make sure that all the chromosomes assort independently. We don't need an anaphase and a telophase. So it's much simpler. If I were to draw it very briefly, your textbook has several steps. I'm just going to draw it really quickly. The first step that happens is the bacteria copies its chromosome, and the chromosome, I'll just tell you, is actually attached on the inside of the cell membrane. And once replication is complete, chapter 9, one of your favorite chapters, once replication is complete, then basically all that happens is a septum is formed between the two cells, and they're going to divide into two cells. And yes, they will be slightly smaller at first in size than the parent cell, but then they will grow a little bit, and then each of them will also engage in binary fission. Now, the very fastest bacteria, the quickest, our favorite lab rat of molecular biology is E. coli. E. coli completes binary fission approximately every 20 minutes. And that meaning, as long as there are enough nutrients, as long as there's enough water, as long as the temperature is satisfactory, the pH is satisfactory, these bacteria will reproduce every 20 minutes. Now, are you kidding me? Sorry, I'll stay closer to the other side. All right, sorry. So every 20 minutes, they're duplicating, and I need you to remember that one cell becomes two cells, and then 20 minutes later, so at a total of 40 minutes, each of these cells is going to become two cells. So then I will have 4 cells, right? We refer to the amount of time that it takes for the bacteria to go from being one cell to 2 cells, to doubling its number, as the generation time. And I need you to remember the generation time 0, we always start with some number of cells. It's either going to be 1 cell or 10 cells. And then every 20 minutes, that number doubles. Okay. Now, if I just let E. coli multiply by itself like crazy without any interference, and again, as long as there are enough nutrients and everything, that E. coli at the end of a day, of a 24-hour period, that E. coli would make more. After 8 hours, let's start with that. After 8 hours, would be over 1 million cells. And after 24 hours, it would be a number followed by 23 zeros. So the number of bacterial cells, it increases super quickly and exponentially. And so when we are counting bacteria,
and then trying to record our numbers over a period of time. It is much more pragmatic or much more practical for us to use a log scale, a logarithmic scale, the power of 10, rather than an arithmetic scale, 1, 2, 3, 4, 5, 6. So when you count bacteria, you actually count them on this logarithmic scale, and you can plot bacterial growth. And we call that chart, we make a graph, and it has a name. It's called the bacterial growth curve, because the curve is the same for every bacteria. The only thing that changes is, as you'll see, the amount of time on the bottom, and that's going to change the shape of the graph slightly. So we can count the bacteria, and we can plot them on, as I said, what we call the bacterial growth curve. And time is on the bottom of the x-axis. So that would be the independent variable, right? And we usually do this time in hours, not minutes, hours. And then on the y-axis, we plot the log number of bacteria. And I don't care what bacteria it is, the graph will always have the same shape. So I'm going to put 0 here for 0 hours, and then I'm going to do them in increments of 2, 4, 6, 8, 10, 12, 14, 16, 18, 20. Oh, what the heck? Let's go all the way out to 24. There, whole day, right? When I plot this, it's going to look like this. There's going to be kind of off to a slow start. We're going to get a little bit of a swoop, and then you get this, and then you get this. And there's a prettier picture in your book, of course, but this phase here, we talk about what's going on, or what the bacteria are doing, or what their numbers are doing in each of these different areas of the curve, and it's been blocked off, kind of like this. And then we discuss each one of these segments that I have blocked off here. The first phase we call the lag, L-A-G phase. And that is the phase where we said the bacteria are getting accustomed to the media. They are reproducing slowly, mostly just doing binary fission as fast as they can, and their numbers are slowly increasing. This is probably this part of the curve and this part of the curve are probably the most interesting to both scientists and people in industry that are trying to use bacteria to produce compounds that we use for vitamins or pharmaceuticals, or produce alcohol or beer or various products. This phase, second phase, we call the log, L-O-G, for logarithmic, or you'll see it called the exponential phase. Exponential, I can't spell that very well or fit it in there. Sorry about that. And that's because, I'll switch to the pink here, I'm not going to be able to tell the difference. That's because this is where we have the biggest curve, the biggest increase in growth, numbers of bacteria for the shortest period of time. Okay, so this log phase is important, as I mentioned, clinically, because people want to know how fast the bacteria is multiplying, because that will indicate how quickly a person needs to be treated, maybe perhaps even what dose of antibiotics that need to be administered. Again, if I'm a person who is studying bacteria out in the environment, or someone who's trying to force bacteria to produce products for me, I want to keep the bacteria in this log phase, this exponential phase, because this is when they're making all of their byproducts, all their chemical byproducts. And believe it or not, this is when it's easiest for a clinician to kill bacteria, because they are making everything. All their enzymes are working at their capacity. This phase here is kind of leveling off phase. It's called the stationary phase. And that means there are equal numbers of bacteria new bacteria forming as well as numbers of bacteria dying, right? Because clearly the graph is not going to go up forever. Usually we say during the stationary phase, this is where bacteria starting to accumulate a lot of waste products in the solution. And if we don't siphon off the media and provide them with fresh food, the pH is going to change dramatically. The amount of free water might decrease. The amount of oxygen might decrease, and carbon dioxide might increase if it's an aerobic bacteria or a facultative anaerobe doing cellular respiration. It might switch to fermentation when it hits the stationary phase. Taking byproducts that might make things taste bad organic acids that we wouldn't want if we were trying to produce food or a particular vitamin or pharmaceutical. So this stationary phase is something, once the bacteria hit that, usually people want to avoid that. Usually they want to go back into log phase. And then this phase here we refer to as phase, because yes, that's what it is. More bacteria are dying than new bacteria are being formed. So we have, again, it's a log or exponential die-off, but we never draw the bacteria all the way down to zero, right? We always give ourselves a little space here, because if bacteria make spores, for example, and I put a pipette in here and I suck out some bacteria and put them in a fresh solution, then I would get the growth curve all over again. Okay. So this is the curve. Now we'll talk about how people, actually scientists and then not just clinical scientists and research scientists, but also people studying the environment, how they actually count these numbers of bacteria to be able to put them on this curve. And there are different methods of counting, and I would just run through those very quickly. So I'm going to bring us back over here. Because my battery is going down. So it's been a long Zoom morning. Ugh. There, that's a little better. That's right in my eyeballs. All right. Yes, it is easiest to kill bacteria in the exponential phase. And the reason for that is because all of the drugs that we use to kill bacteria or decrease their numbers, they work best when the bacteria are producing the enzymes or products during their metabolism, because we usually use metabolic products that we're targeting to kill the bacteria. So think about bacteria making cell wall, for example, right? We want to target when the bacteria is making the most cell wall that we can do the most damage, not when they're just sitting there, just doing their metabolism, not actually growing. All right, so now let's look at some methods for counting. And I'm going to put up a whiteboard and draw some of these out for you, poor or not so poorly. So first, just going to type. So this comes under many names. Sometimes you'll see it called enumeration of bacterial growth. Sometimes you will also see it measurement or measuring bacterial growth. All of those are, used interchangeably. Okay, so one method that is used super common is called the, it was number one, serial solutions plus, okay? And the plus is either a pour plate or a spread plate. So with pour plates, I should say pour plates or spread plates, right? I'm going to make another text box and put the second method that we're going to discuss underneath that. Then we're going to go back. Another method would be to measure turbidity. And for that, we're going to need to use a spectrophotometer.
And that's important because we're going to talk about spectrophotometers later in the semester, and it will be helpful if you have a little understanding of what a spectrophotometer can do. And then the third method here is just physical counting, called direct count. And for that, we're going to use a chemocytometer if we're counting it on a slide, or some sort of device similar to that. That's counting chamber. And then we're going to either use a Coulter counter or a derivation of that, which is known as a fax machine, but not like you think of fax, FACS, which stands for fluorescent activated cell sorting. Okay, so we're going to go through these different methods of counting here, 1, 2, 3. All right, so let's start with serial dilutions and pour plates. I think I'll probably go back to the chalkboard for this actually, because I'm going to have a hard time drawing this with my right hand. So I'm going to stop that share and back over here, erase. Okay, so for serial dilutions and pour plates or spread plates, I'm going to demonstrate spread plates, but you should be aware that you can do a pour plate as well. And I'll tell you a reason why you might want to do that if you, like, again, if you were out in the field. So first part of this is you have to make a serial dilution. So what that means is a dilution in series. You usually choose the same dilution factor. So I'm going to choose 1 to 10 as the dilution factor because that's simple math. But it could be a 1 to 5, it could be a 1 to 20, it could be a 1 to 400, depending on the application. So here's my sample. I'm going to put it in a flask over here. Right. So let's say I test water, and I usually go out and I'm testing for, I don't know, bacteria that can make people sick, give you diarrhea, E. coli, or maybe I'm testing for cholera, or maybe I'm just testing for all kinds of things that could be tested for. But most likely there are going to be too numerous to count bacteria in that sample, which is the whole reason that I would need to dilute my sample. Okay, so I'm going to collect my sample, and then I'm going to I'm going to draw a few test tubes here. I'll draw 5. And into the test tubes, I'm going to have some liquid media, some broth. And I'm going to take, let's say I have 9 mils of broth in each of these tubes. Okay. And I'm going to take a 1 ml sample, a 1 milliliter sample from my, what I harvested from the environment, from my water sample, or maybe I'm checking pasteurization of milk. So I'm going out to various farms, dairy farms, and I'm checking their pasteurization. So I might have a milk sample. So I'm going to put that 1 mil into the 9, which is going to produce a 1 to 10 dilution, 1 mil in 10. And the reason we call these serial dilutions is because I'm going to repeat the 1 to 10 dilution, but I'm going to take a 1 to 10 dilution of my original 1 to 10 dilution. And so the series means that every time I dilute, I'm going to be multiplying by that same dilution factor.
So I've got a 1 to 10 dilution here, and then I'm going to take 1 ml out of that solution and transfer it to the next. And that's going to give me a 1 to 10 times a 1 to 10, which is a 1 to 100 solution. And I'm going to do it again and again and again. And you can see I'm going to get 1 to 1,000, 1 to 10,000, but I'm going to switch to scientific notation here because I can't write out all the zeros. And then 1 to 100,000. 1 times 1, 1 to 1 times 10 to the 5th. So that's the serial dilution part. And then for each of these tubes, I'm going to take a sample and I'm going to plate it on a nutrient agar plate or some sort of nutrient media that the bacteria will grow on. And then I'm going to wait until tomorrow, and then I'm going to count how many colonies grew on my plate. Every one of these dots will be a colony. And they should decrease in number as they get more dilute. The first few are usually what we say too numerous to count. And those would literally be described as TNTC, too numerous to count. We are looking for a plate that has colonies between 25 and 250 colonies. And the reason for that is because statistically, we have figured out that less than 25 colonies usually is an undercount, and more than 250, usually the scientist or lab technician has a hard time actually counting all 250 colonies accurately. So we're looking for a plate that has between 25 and 250 because it's manageable and it will be statistically significant and reproducible reliably. And for me, that's going to be this plate here. You may not be able to see, but I put some dots on here. They're very small. I realize that, but what I have here. I put about 25 dots on this column, on this plate. Actually, let's say there's actually 32. No, let's say it's 41 dots, 41 colonies. So then all I have to do is I take my 41 colonies and I multiply it by the dilution factor that originated those colonies. So in this case, it's 41 times 1,000. And so I would just add 3 zeros here, which would give me 41,000 colony forming units per milliliter. Colony forming units able to make a colony per mil, because remember my dilution factor is 1 mil, and I transferred 1 mil from my test tube and put that on my Petri dish. Now, the difference between a pour plate and a spread plate, a pour plate, you would do the same procedure, except rather than using broth here, you would use melted agar, and then you would pour that melted agar into an empty, sterile Petri dish. If you're doing a spread plate, literally, you would use broth, and you would put, with a pipette, you would transfer your 1 mil onto the Petri dish, and then you would spread it around. That's why it's called a spread. Okay. This is super common method for counting bacteria. You can decrease the amount of volume in these test tubes, which will increase the dilution factors. It can be made more portable by using very small tubes that are self-capping and then plating your results on small Petri dishes, literally that are only this big, as opposed to the ones that you have in your refrigerator that are normal size. They're like 60 millimeter size. We can get them on smaller ones. So this is very frequently the way people count bacteria. Now, if you were trying to make that bacterial growth curve, which I erased, you would have to set this up every hour, right? Or every 2 hours in order to plot those numbers of bacteria. That's ooh, that's a lot of work. I don't know about you, but I would rather find an easier way to count, right? So if you guys refer back to the slide that I shared with you earlier, the second method, I believe, was turbidity that we were going to look at. And I am going to have to draw on this side of the board. This is an approximate method, but it is a really good approximate method because it is based on hundreds of these that have been done with a particular bacteria to form a reference chart that you can use to look up a really good approximation of how, remember, turbidity is how cloudy is your solution. Cloudy indicates there's bacterial growth in the solution, and we can use this device called a spectrophotometer that can actually quantitate the cloudiness or the turbidity. It's really cool. So I am going to erase some of my dilutions here. So I have a little more space. I don't want to completely erase them because, as we're going to see, sometimes you have to do this first, even before you can use the turbidity method. Now, the turbidity method involves a device that we call a spectrophotometer. Which is a machine of various complexities. Now they're all digital, of course, but the picture in your textbook looks like a Spec 20, which you might have used in chemistry. And the guts of the machine are what's important, okay? So I'm just gonna draw those. So basically what the machine consists of is a light bulb. And it's a fancy dancy light bulb that sometimes can be emit ultraviolet light and sometimes does white light and sometimes now can even do infrared. And I'm going to draw two of these light bulbs because I'm going to show you two different scenarios with the spectrophotometer. Next to the light bulb or after the light bulb, there is a chamber, and it usually is actually rectangular. And into that chamber, you can only fit a very specific tube, test tube, spectrophotometer tube, because it's made of plastic or glass that is going to have a matching refractive index with the light bulb, so we get maximum light through our specimen. Now, I'll try to draw the bacterial colonies a little bit larger this time on my picture. So in the case here where I have clear broth and zero bacteria, right? And in this case, I'm going to have some number of cells inside of here. So yeah, you can see those. So I'm just drawing them, exaggerating how large they are, right? So those are some bacterial colonies floating in the broth. This one is empty. This is my blank. So in this case, where there's zero bacteria, the light rays are predominantly going to travel directly through the solution, and they're going to strike a detector. I'm going to draw here like this. And this detector, you can think of it, originally they were magnetic, but now again, they're digital, but I'm going to draw an old-fashioned kind where on the right-hand side here, I'm going to put 100% T for transmission there, and over here would be 0% T, and this of course would be 50, this would be 75, and of course this is a gross, minimization here. These numbers could be a lot more accurate. And basically there's a dial. So the more light rays that hit my detector, the more they are going to move a needle here. So that needle is going to point at 100% transmission, or all the light rays are transmitted through the solution to the detector, and that is reflected by this 100% transmission. So this is clear broth, no bacteria. I think some of you can kind of see where I'm going with this, right? If on my other situation here, this is not working. I have to switch to blue. If here I have another detector, another spectrophotometer chamber, and I have my 100% transmission and my 0 and my 50% and my 75% and my 25%, right? And again, I have that little needle. Now the light is going to come into this solution, but oh, it's going to hit some bacteria because there's bacteria in there. The light will be reflected and refracted. So fewer of the light rays are going to make it, I should have made it so it doesn't hit any bacteria, all the way over to this other side. More of the light rays are going to be bounced around inside. So I have fewer light rays being transmitted through the solution, which is going to impede my detector here. And let's say now I only have 50% transmission. And you're like, okay, so what does that mean? Well, what that means is because other scientists and researchers have conducted this experiment every half hour with bacteria, and this experiment as well, they know exactly, for example, if you started with a solution that had 10 E. coli in it, they can tell you that they made a reference chart. So at 2 hours, for example, or 4, let's make it 6 hours, I can just take a sample, maybe run some serial dilutions, and then rather than plating it out and waiting till tomorrow to count my colonies, I can just take a sample of this, pop it in the spectrophotometer, and whatever number this is, then I can look it up in the table and it will tell me with a pretty good approximation, 2 hour, 4 hour E. coli sample with 50% transmission is approximately equivalent to 382,000 cells, colony forming units per mil. So although it seems like a lot more setup because I have to have the spectrophotometer, I think you guys can appreciate if I'm looking for a bacteria that we already know what the reference chart is, or if we take the time to make the reference chart for the bacteria, then I only have to go through this whole process once or twice, right? All from start to finish, every hour, every 20 minutes. And then I can just do a cheater. I can just get a really good really close accommodation or a really close number based on how cloudy is the solution. It's a huge time saver. And again, I'm going through this and emphasizing it because the spectrophotometer is going to come back to haunt us later. This is how exactly same machine that is used to detect how much antibody you have for pretty much anytime an antibody titer is conducted. Basically your antibodies, your blood is drawn, the serum is separated, and then they look for how much antibody you have, say to the measles virus, and this is how they do it. They do it by a spectrophotometer, how many antibodies are clouding up the solution, right? So this will come back and haunt us later. All right, I'm gonna leave the spectrophotometer up there and just erase this part, the serial dilution part. And our last method of counting is actually just counting, direct counting. And I wrote down hemocytometer there because that involves a special slide, super thick, thicker than any glass slide you've ever seen. And in the center of the slide, etched with a laser, is a tiny, tiny grid that looks like a piece of metal when you look at the slide. Once you tip it up to the light, you can maybe see the fine lines. And it has a special size. There's a little special gap in the glass here. So you can load 10 microliters. So remember, a microliter is 1/1000th of a milliliter. So you can load 10 microliters onto this slide, put the cover slip on, and then you can actually see bacterial cells in the grid and count them. It involves a lot of multiplication for your various dilution factors, but once you get good at it, it's pretty quick, right? I see you can't see that microliter symbol there, right? It's a mu, microliter, not an M, a little Greek mu. So, and then this is fine. It's time consuming, right? And you have to clean your slide between each one because you don't want any cross contamination. And so your book shows what we call a Coulter counter. This is actually designed to count red blood cells and white blood cells. And I like it because it's a very simplistic picture that they've given you. We've got a funnel here basically where you can load a solution and a piece of tubing that the cells can only pass through that tubing in single file. And then there is a magnetic field along the tubing. And as the cells pass through the magnetic field, the machine counts for you. Okay, that's what Coulter counter is for. It's awesome unless this piece of tubing gets clogged. And then it's a little tricky because then it doesn't work very well. The modification that I mentioned that we call a FACS machine, FACS, fluorescent activated cell sorting, is a modified Coulter counter where at the end of this piece of tubing, we may have multiple test tubes. It's a little bit bigger device. Let me erase my hash marks there for my counting. So these are supposed to be tubes. Fluorescent activated because rather than just counting the cells based on their size, we count the cells based on what markers they have on their surface. So for example, if we're trying to separate helper T cells from cytotoxic T cells, helper T cells are usually CD4 positive, and cytotoxic T cells are CD8 positive. If you're trying to figure out, for example, if an HIV patient or a hepatitis patient, what complement of CD8 versus CD4 cells, you can label the cells with different colors. So I'm going to label the CD4 cells pink. I know you can't see very well, but let's do pink and blue because the blue kind of shows up. All right, so let's say I labeled the CD8 cells blue and the CD4 cells pink. The fluorescent activated cell sorter, rather than a magnet here as the detector, it's a laser which excites the dyes on the surface, and then it sorts them into tubes for you. It not only tells you how many, it can actually sort the pink ones into one tube and the blue ones into another tube. This is really, really useful, especially if you are trying to study a person's immune response to a particular infection, for example. And I'm going